Welcome back to The Explainer. Today, we're unpacking a medical and veterinary nightmare that has literally haunted humanity and our animal companions for centuries. We're looking at tetanus. It's a disease so intense, so visually striking, and honestly, so devastating that it's practically impossible to forget once you've seen it. Over the next few minutes, we're going to trace the exact journey of the incredibly resilient Clostridium Tetani bacterium, from just sitting harmlessly in the dirt on the ground to triggering severe, agonizing spasms in both humans and horses. Okay, let's dive right into this. You've probably heard the old warning a million times. Step on a rusty nail and you're gonna get lockjaw. But is it actually the rust that causes the disease? Well, it turns out the rust itself is totally innocent. It's been framed. The real truth is a fascinating biological mystery about an invisible, environmental survivor that exploits a very specific type of wound to unleash one of the most potent neurotoxins known to science. So here is our roadmap to understand this disease today. First, we'll meet the bacterium. Then we'll map out the neurotoxin sabotage, look at the clinical reality of spastic paralysis, explore the clinical puzzle of laboratory diagnosis, and finally, wrap things up with treatment and prevention. Starting with Section 1, meet the bacterium Clostridium tetani. The absolute foundation of understanding tetanus is understanding its relationship with oxygen. Clostridium tetani is an obligate anaerobe. Put simply, it entirely lacks the biological enzymes needed to survive in oxygen. Oxygen is literally toxic to it. And you know what? That perfectly explains the rusty nail trope. If you get a superficial scrape on your skin, it gets plenty of oxygen, so this bacteria simply can't thrive. But a deep puncture wound from a dirty nail, that creates a low-oxygen, necrotic pocket deep in your tissue. It is the absolute perfect breeding ground for these bacteria to wake up and start multiplying. If you actually peek at this bug under a microscope, it has a very classic visual buzzword. It's a gram-positive rod, but when it forms spores to protect itself from the environment, the spore develops right at the very end of the cell. This creates a distended spherical terminal tip. Microbiologists universally describe this as a drumstick or tennis racket morphology. That little round spore at the end, that is its biological armor. And let me tell you, that armor is incredibly effective. These spores are ubiquitous, meaning they're literally everywhere. They hide out in soil, dust, animal feces, you name it. They can survive in the environment for years, easily resisting intense heat, boiling, and even standard disinfectants. They just sit there, waiting. In humans, they typically enter through deep puncture wounds or, tragically, through the infected umbilical stumps of human neonates born in unhygienic conditions. In horses, something as simple as a hoof abscess or a castration site can be the perfect doorway. Section 2, neurotoxin sabotage and pathogenesis. So what actually happens when they get inside? The terrifying truth is that the bacteria itself doesn't actually invade and destroy your deep tissues. Nope, it just sits in that local wound and manufactures a weapon. Here's that precise pathological journey mapped out for you. The spores enter the deep wound. Finding that nice, anaerobic, oxygen-free environment, they germinate into active bacteria. Then they produce and release their toxin. Now, rather than just floating aimlessly in the blood, this toxin actually enters the local peripheral nerves and travels backward, a process we call retrograde transport, sneaking all the way up the nerve fibers directly into the central nervous system, where it executes its primary sabotage. That weapon has a name: Tetanospasmin. Down in your spinal cord, you have these things called Renshaw cells, and their job is to release inhibitory neurotransmitters, specifically GABA and glycine. Think of these as the off switch for your muscles, telling them to relax after a contraction. Tetanospasmin aggressively blocks the release of these inhibitory signals. Without GABA and glycine, your motor neurons completely lose their brakes. The result? Continuous, unopposed muscle contraction. Section 3, spastic paralysis and clinical features. The physical manifestation of this disinhibition leads directly to a terrifying clinical fallout. The earliest classic sign is usually trismus, which is widely known as lockjaw, caused by the uncontrollable spasm of the powerful masseter muscles in the jaw. As it spreads, patients develop risus or sardonicus, this eerie, forced grin caused by facial muscle spasms. The most dramatic sign, though, is opisthotonus, where the back muscles spasm so violently that the entire body arches rigidly backward. And because the nervous system is so hyperexcitable without its inhibitory controls, these agonizing spasms can be triggered by literally the slightest stimulus: a quick flash of light, a sudden noise, or even just a light touch. But humans definitely aren't the only ones who suffer from this. According to the AAEP Infectious Disease Guidelines, horses are exceptionally sensitive to tetanus neurotoxins. A horse with tetanus will often display a wide-based, rigid sawhorse stance. They'll exhibit an extended neck, an elevated tail, and a very characteristic protrusion of the third eyelid. Just like in humans, the mortality rate is staggeringly high, often reported at well over 50%. And this brilliantly illustrates a classic medical comparison that students are always tested on. Both tetanus and botulism are caused by Clostridium bacteria, but they do the exact opposite things. Tetanus decreases the inhibitory signals, causing tight, uncontrolled contractions, so spastic paralysis. Botulism, on the other hand, stops the excitatory signal, acetylcholine, causing loose, floppy muscles, flaccid paralysis. It's essentially a perfect mirror image of neurotoxin sabotage. Section 4. Laboratory Diagnosis, a Clinical Puzzle. So, when doctors or veterinarians suspect tetanus, how does the lab actually confirm it? Well, according to the Public Health Laboratory Network, isolating this bug is incredibly tricky. Because it's an obligate anaerobe, culturing it requires strict oxygen-free environments. And even when successful, after 24 hours of incubation, Clostridium tetani only produces this really thin, highly transparent film of swarming growth on the agar surface. It is notoriously easy for lab technicians to completely miss this fine film unless they know exactly what they're looking for and physically track across the agar with a loupe. Therefore, the diagnosis pretty much remains primarily clinical. But if a lab needs absolute, definitive proof, they turn to the rather fascinating mouse toxicity test. They take a liquid culture of the suspected bacteria and inject the filtered fluid into the thigh of a small mouse. However, because the bacteria only releases the toxin as it naturally breaks down, a process called autolysis, the culture might need to sit for up to 4 days before there's enough toxin to even detect. If the mouse develops a stiff, paralyzed leg, and if a second control mouse protected with tetanus antitoxin remains perfectly healthy, the lab has its smoking gun. The toxin is definitively identified by biological neutralization. Section 5. Treatment and Prevention Strategies. All right, so once diagnosed, how do we actually fight back? The treatment for tetanus is a massive, aggressive, multi-pronged counterattack. First, doctors must physically clean and debride the wound to eliminate the breeding ground. They administer antibiotics like metronidazole to kill off the active bacteria. Crucially, they give human tetanus immune globulin, an antitoxin that acts like a sponge, neutralizing any toxin still floating in the blood before it can reach the nerves. To manage the horrific pain, strong muscle relaxants like benzodiazepines are given. And finally, the patient is placed in a dark, padded, and extremely quiet room to prevent any sensory stimuli from triggering further spasms. Now, in veterinary medicine, specifically for horses, there is a very important warning regarding that antitoxin step. The AAEP guidelines note that there are currently no tetanus antitoxin products officially labeled for horses. Administering TAT to a horse represents an off-label use and carries a distinct biological risk, the potential transmission of equine parvovirus, which causes a dangerous liver condition known as Theiler's disease. Vets really have to carefully weigh this risk when treating or trying to passively protect an exposed horse. Let's lock in everything we've just covered with a rapid review mnemonic: TETANUS. It triggers spasms, causes extensor rigidity, the toxin blocks inhibition, it's an anaerobic gram-positive rod, it utilizes a potent neurotoxin, leading to uncontrolled contractions and ultimately spastic paralysis. If you can remember that list, you understand the absolute core pathophysiology of this disease. Here is a wild fact to leave you with. Surviving a natural tetanus infection does not provide your body with future immunity. The amount of toxin required to kill you is actually smaller than the amount required to trigger an immune memory response. The only way to truly protect yourself and your animals is through the toxoid vaccine, which is incredibly effective, because these spores are hiding out in the dust right outside your door right now. When was your last booster? Thanks for joining us on this explainer. See you next time.